And in looking at this lecture, one of the things we're going to be doing is seeing what anatomy and physiology is all about. So, uh, of course, anatomy and physiology is talking about the structure and the function of the body. So we are going to look at structures such as the skull or your intestines, but we're also going to look at what do they do? What is their purpose? And so as we're looking at this, we know this is an advanced study for those of you who are thinking about going into a healthcare career, exercise physiology, pathology, and some of you are taking this because you are just interested in the human body. So when we think about studying our form, we have to think about how do we do that? How do we go about looking at the form of a human body? Well, one of the first things they ever did just to figure out form was dissections. And so there were people who were artists who wanted to know more about the form. So they would look at cadavers to find out the form. Our early physicians did the same thing. They would open people up. They would look inside of them. They would try to figure out what was going on with them. So dissections was one of the ways. Since about the 1700s, we've been doing medical imaging. X-ray came around about that time. And so what we're doing is we're going to view inside of the body without opening it. So radiology, of course, is a branch. There are many different parts of radiology. We just have x-rays where we actually pass a beam of x-ray through the body and we can see different parts. There is, of course, CT scanning and MRIs, echo scanning, and some others. So if you are interested in medical imaging, that might be one of the things you think about. This, of course, is a medical image. Now, another thing that doctors do every time you go in to see them is a gross anatomy study of you. So gross anatomy means you just look at the person and you look at everything from their skin to their eyes to what their nose looks like, what their feet look like, and you'll be trying to figure out some things, such as if I came into my doctor's office and my hands were purple, one of the things they would be trying to do is figure out why, and quickly, by the way, because that means there's no oxygen getting to them. So everybody does a gross anatomy study, whether you want to or not, every time you look at somebody. Now, histology is another way. Now, this is microscopic anatomy, and histology, you're just studying the tissue, and you must have a microscope to do this. Another one is cytology. This is where you actually study the cells inside the tissues. So you're starting to look at how cells are they the right size? Do you have the right number of them? And their shape. So those are some of the things you're going to do in cytology. Now, physiology, on the other hand, is the study of the function. And so how does it work? It does look at both molecular and cellular levels of a human body. And we're looking at things like, is it absorbing water correctly into the kidneys? Is it digesting the food that goes into the stomach? Are you having thought processes that help control the rest of your body? So there's lots of different things we look at when we study the physiology of a human body. So as we're looking at the human body, we have to think about its complexity and how does that complexity coincide with maybe an anatomy issue or a physiology issue. So we start with the largest part here, which is the organism itself. So you're looking at a human body. You then can break it down into its organ systems. So when we're looking at the human body, there are 11 organ systems, and according to some new scientists, 12 organ systems that we'll talk about later. But you look at all the different organ systems, such as the digestive system, the urinary system, the nervous system. So you look at all these different things. Then you break it down again into a smaller component, which is your organs. So if you're talking about the urinary system, you may just talk about the kidney. Or if you're talking about the digestive system, you may just talk about the small intestine. Or if you're talking about the nervous system, you may just talk about the brain. So again, we're breaking it down into smaller components so we can look at it. So organs then are built of tissues. And tissues are basically just a group of cells, which is our next step down. So we'll look at cells. And then inside of each cell, you have organelles. And these organelles are small, but they fit inside that cell. And then from the organelle, you go to a molecule, and then from a molecule to an atom. So if I was going in this hierarchy from largest to smallest, I would say organism, organ system, organ, tissue, cell, organelle, molecule, and then atom being the smallest part. So as we're talking about the human body, we have to talk about it being stable. How do we keep the internal conditions stable in the human body? When we talk about this, we talk about its homeostasis. Homeo meaning same, stasis meaning stability. So you look at how are these conditions staying stable. One of the conditions you might think about is the human temperature. Okay, so the body temperature. It is 37 degrees Celsius normally, but you need to see is it being stable? Is it staying at that 37 degrees Celsius, or has it dropped to 34 degrees Celsius, which means you're in hyperthermia? Or hypothermia, I'm sorry. Or did it rise up to about 39 degrees Celsius, which means you're in hyperthermia? So again, homeostasis, you got to know what the normal internal condition is before you can decide whether or not it's out of homeostasis. Well, one of the ways we bring it back into homeostasis is through negative feedback. Now, negative feedback senses that the body has changed and wants to reverse it back to normal. So whenever we are running a temperature, one of the things that happens is we begin to sweat. Okay, so we're sweating to try to cool the body. Or if we're too cold, we start shivering to try to warm the body. Again, whenever we see a loss of homeostatic control, it could be caused by illness. So it's one of the things to be thinking about as we're thinking about homeostasis. So we're going to talk about this negative feedback loop. So if you wanted to keep the body at 37 degrees Celsius, one of the things that we know is that your hypothalamus is going to detect when your temperature goes up or goes down below that norm. Let's say it goes up. If it goes up, the hypothalamus then connects to your brain and tells the brain, okay, we're too hot. We need to do something to cool off. And so you begin to sweat. Then your body temperature begins to drop, and it may drop too much. So if it drops too much and you begin to chill, you begin to shiver. Think about somebody who's been sick, how that is a common, normal reaction to having a temperature. So in this negative feedback loop, you have three parts. You have the receptor, you have the control center, and you have the effector. Now, the receptor is the structure that senses change in the body. So again, it's going to understand that there is a change in the body. Then the control center is the one that makes the decision and says, hey, listen, we need to fix this. And then the effector is the cell or the organ that carries out the instructions from the control center. So let's go over the temperature one again. So my receptor in this temperature is the hypothalamus. That's my receptor. The control center is our brain, and then the effector is our skin releasing fluid to help us cool down. Now, opposite of negative feedback is positive feedback. Now, positive feedback is different in the fact that it doesn't go through this slow cycle, but it goes through a very rapid change. And so, one of the things about positive feedback is that it is self-amplifying. So instead of doing this little slow fixing of it, it actually enhances whatever the effector is, and it enhances it quickly and enhances it uh, logistically. So in other words, it does it really quickly. So it is going to make very rapid changes in things like, think about contracting for childbirth. That's the one that most people use for this positive feedback example. So when you first start contractions, they hurt a little bit, and you're doing some motion, but by the time you get to the end to give birth, you are having very rapid contractions and pushing really hard to get the child out. Another one is blood clotting. Blood clotting doesn't happen slowly. If it did, we could bleed to death before it happened. So blood clotting happens very rapidly. As soon as you cut yourself, you begin to build a clot. That's how it works. It can be very dangerous to do this positive feedback loop. Think about blood clots gaining in places they shouldn't be. So that's one of the things you have to think about with positive feedback. So we're going to talk about something about chemistry right now, and what we're talking about is gradients. Now, gradients is a difference in chemical composition, or a difference in temperature, or a difference in pressure. And what happens is matter and energy both tend to flow down a gradient, which means that wherever the greatest chemical concentration is, the greatest temperature, the greatest pressure, it tries to make it flow to the lowest point. So if I have a lot of potassium in my body, it's going to try to flow to some place that it needs to be, as opposed to where it is at the beginning. So if I've got potassium on the outside of my cell and I have very little inside my cell, it's going to flow into the cell. Another example is blood pressure. So if my blood pressure is really high close to my heart, which it normally is, then it's going to push that blood or run down that gradient towards my toe where the blood pressure is really low. So again, think about gradient and flow. So again, we're going to talk a little bit about medical imaging. First thing I want to tell you is that I worked in medical imaging for several years, so I know a little bit about it. And we look at how to take pictures of the inside of the human body so we don't have to open them. X-rays are used to penetrate the body so that we can see certain structures, such as bone and teeth. You can actually see tumors on an X-ray. They do amount for about half of all medical imaging. To make things show up like your stomach or your esophagus, because they are not as thick as bone. So you want to be able to see them and see how they are working. You may have someone either swallow or have an injection into the body so these hollow structures fill up and you can see them, such as having a barium swallow. The barium is a very dense, opaque chemical, and so when you swallow it, you can actually see it going down. Or you may give an injection so that you can see blood vessels in the body on a CT scan or on an MRI. So CT scans are low intensity x-rays. So they are still x-rays. And what they do is they take images by not looking at you anterior to posterior, but looking at you from superior to inferior. So they take what they call slices of you, and what they'll do is they'll start taking an image here at the top of your head, and for every 1 millimeter, they'll take another image. That's how it works. The nice thing is, because they do it that way, you get a very sharp image of what is in that particular part of the body. With MRIs, you are not using x-rays anymore. Now you're using magnetic fields and radio waves. And so, again, they do the same type of imaging as CT scans, going from superior to inferior. But instead of just looking at structure, you now can look at function, because it gives off a different level of this magnetic field and radio waves, which are slower and will give you a better image of what is going on in the function of that particular organ. There is another type of medical tomography called PET scan. And in this one, you actually are looking at all the chemical reactions that are going on inside of a tissue. So with this one, what you do is you inject a radioactive isotope with glucose on it. And the reason why you use glucose is used in every cell. So it's going to go to those cells. And then you start looking at positrons that are being given off. And as you're looking at that, you can see where most of the glucose goes to. So if it's a cell that's rapidly dividing, you're going to get a lot of glucose in there. Tumor cells rapidly divide. Anything that's not dividing and is damaged will show up darkly, and so you don't get to see anything from it. Now, sonography or echography is the second and oldest type of imaging, and it uses high frequency sound waves that echo back towards the machinery to give you an image. We did this because we wanted to avoid any harmful x-rays in obstetrics or childbearing. And the other thing was we wanted to look at images of something that we had not seen before. Now, the problem was with the beginning sonographies is they were this gray and black picture that just had a bunch of dots on it, and it was really hard to tell what was there unless you were very well trained. However, today we now do 3D imaging, and as you can see in this picture here, you can tell that's a baby with no problem. You see the eyes and the eyelid, the nose, the mouth, the cheeks, the arms, the hands. So this has come a long way in the last 30 to 40 years. So I hope you enjoyed this beginning of our anatomy lecture, and I will see you next week.